Okay, once more. This time with feeling. Yes? Okay, thank you. Okay, so I'm Alex Bott. I work with Jared Rutter at the University of Utah. And I want to thank the organizers for the opportunity to present today. I registered for this to learn, not necessarily to present. So it was a little bit of a shock when I got the email that said, hey, you're a cell biologist. Why don't you tell us about what you're up to? So a little bit intimidating, I think, especially following Aviv last night and Marion today. I think I'm going to be the first person not to talk about perturb-seq, but I'll tell you about a different way that we want to try to tackle this problem. So I have two types of opening slides, one geared towards biologists and one geared towards everybody else. So I'm giving you the everybody else because this is how a biologist like me thinks about gene regulation. In general, a cell will respond to a signal. So that could be some kind of ligand that you add, a small molecule, a perturbation to metabolic space, something that induces some change in the cell. That signal is transduced by a signal transducer. And maybe that culminates in a transcription factor that becomes activated, and it binds to DNA. It turns on a gene expression program. And then all of the fuzzy stuff, right, that I think this group does not need to be elaborated, but there is chromatin level changes. There's accessibility that opens and closes. There's looping. There's all sorts of exquisite regulation that occurs in order to produce an RNA transcript. After the transcript is made, you still have to splice the RNA. It has to be exported from the nucleus. It can be degraded. So all of this is kind of wrapped up into a simplified version and prompts the question that I wanted to set out to answer, again, as a cell biologist, is how can one identify the set of factors that control a gene's expression? And I wanted to do this in a very gene-centric way because when I joined Jared's lab, the question I wanted to tackle was, if I perturb metabolism, if I remove a nutrient, how does the cell sense that, right? And so we figured if we want to ask a question about gene regulation in an RNA molecule, we should start there. And if we start with an RNA target, can we measure it? And the answer is yes. So we can use fluorescent in situ hybridization. So we borrowed some chemistry from Brian Bellevue at the University of Washington to do this very cheaply and with high sensitivity. So we designed DNA oligos that tile across an RNA of interest. And they leave this kind of tail open that acts as a landing pad for fluorescent secondary molecules. So now we can visualize or measure in a quantitative way that RNA abundance. And if we can do that by flow cytometry, we can put a CRISPR screen on the front end. And importantly, what we really care about is the ability to not just perturb genetically, but to add a second layer, right? So we can, every cell gets a unique genetic perturbation, but also we can ask a question, how do they respond in the presence of this drug, right? And if we can, infect with a CRISPR library, give some kind of perturbation, measure the target molecule, we can sequence the guide RNAs, and we can read all of that out to hopefully understand how transcription is regulated for that given gene, right? So targeted readout to understand transcription, or we call it troutfish for short. And the idea here is it's very deep data on one given target, right? So we're not doing the perturb-seq view where we're asking across thousands of transcripts and thousands of perturbations, but we're able to go to whole genome scale with relative ease for a set of RNA molecules, right, for a set of targets. Okay, so just as a proof of principle, we took the small molecule melton-3A, which is an inhibitor of the MDM2 E3 ligase. Normally, it binds p53 and degrades it. If you treat with this small molecule, that degradation is prohibited. p53 forms a tetramer, goes to the nucleus, and turns on gene expression. And so if we treat cells with melton-3A, we get stabilization of p53, activation of p21, and importantly, at the transcript level as well, right? So schematically, what are we seeing? So in the vehicle treated, there's just less transcripts. It's really a simple assay, right? In the melton-3A, we know p53 activates CDKN1A, that's p21, and it's just more transcripts, which means more green dots, more fluorescence, right? And we can measure that here. So if we run this assay and we just measure by flow cytometry, in the pink or the blue, you see the vehicle or the inactive enantiomer respectively, and the green, you see that nice rightward shift. And so now knowing this, that we can measure a quantitative readout for our target RNA, what if we put the CRISPR screen up front? Can we catch the regulation of p21 in response? And the answer is yes. So we use the transcription factor specific library, and the only hit that we get back is p53, essentially saying that this model can catch that delta, right? So we can ask the question of how is a given gene regulated in response to some stimulus? Okay. We could do the clean genetic knockouts. If we delete p53, obviously you can't turn on p53. You can't activate p21, and there's no change at the flow level. So we had this system in hand, and when I started in Jared's lab, I wanted to ask and answer questions about nutrient sensing, but we kind of pivoted a little bit because we've gotten really interested lately in a super cool metabolic pathway, glycolysis. And it's really interesting for a couple of features. One, it's one of the most ancient metabolic pathways that exist. It's conserved in just about every organism that we know. And if you think about it, it's really elegant, right? There's unique enzymatic function at every step of this coordinated pathway. So everybody works together with its teammates, right? With the goal of transmitting carbons in the form of glucose to pyruvate. But at the same time, there are unique gene products with concerted metabolic effort or enzymatic function. And so I don't have time to tell you about this today, but this implies inherently that there is unique regulation. So you have PFK, PLNM, platelet, liver, and muscle isoforms. That inherently tells us that there's probably some interesting regulation about those. So to study this, we took 2 cell lines, a skin line, Mewo cells in orange here, and K562 in blue. The skin line is highly enriched for glycolytic genes. The K562 is kind of middling expression for most. And importantly, none are, the high expression of glycolytic enzymes is not due to copy number amplification. So if we run our troutfish assay, it's good that we can catch knowns. So we know that MYC is a master regulator of glycolytic metabolism, and we can catch that here. We can do the validation, and that's great. But what we got excited about is that when we run these assays within the lineage, we see some cool stuff pop out. So here, just looking at 2 glycolytic genes, we see SOX10 and MITF in the skin cells. And in K562, the erythroleukemia-like cell lines, we see KLF1, NFE2, MIB, and GATA1, suggesting that perhaps there's a metabolic, or there's a control of metabolism that's encoded by lineage-specifying transcription factors. And if we make the CRISPR-I, if we suppress SOX10 or MITF in the skin cells with CRISPR-I, we can see that melanogenesis genes go down. That's a nice positive control. So these are genes that are part of the specific organelle that only exist in melanocytes. But we see that glycolytic gene expression also decreases. We can do stable isotope tracing, so we can feed cells with C13 glucose. This is slightly heavy. It's stable isotope, not actually radioactive glucose. And we can measure what the cells do with that glucose. And so here you can see that if you suppress SOX10, you have less lactate production. This N3, it's an isotope of lactate. And we can see that all the intracellular intermediates are also diminished. So now we have this question that through our screen, we could identify lineage-specifying transcription factors control or suggestively control glycolytic gene expression. We can see that we can see the metabolism output. The question is this direct? So we tested tons of SOX10 antibodies. They're all kind of bad. But we landed on some MITF antibodies that worked really well for ChIP-seq. And so when we chip MITF, we can see at canonical genes, as it should be, TYRP and DCT, there's melanogenic genes. But in fact, at every glycolytic gene that we looked at and that we ran our troutfish assay on, we can see that MITF binds here, suggesting that there's a direct link here, that it's not just an indirect regulation, but perhaps a direct binding. We wanted to know, is this generalizable? Can this go beyond just the 2 cell systems that we looked at? And so we've started to answer that question by utilizing extant ChIP-seq data. So we went to ChIP Atlas. So this is reprocessed all ChIP-seq binding data in the public domain. And we could just query glycolytic genes and metabolic genes and ask in a given lineage, like in a neural lineage, MYC-N, or in blood, lung, et cetera, do those lineage-defining transcription factors bind to glycolytic genes? And the answer is yes. And we're trying to look at this more systematically. And so if we take all ChIP-seq data across the entire public domain and ask, is there an enrichment of lineage-specific transcription factors binding to metabolic genes? The answer is yes. And to kind of close the loop, we wanted to know, okay, if metabolic genes are regulated by lineage transcription factors, what is the so what? Why is that important, right? And so we wanted to test the hypothesis, if you perturb metabolism, right, which is this intermediate link, does that affect the lineage fate, right, or the lineage function? And so here we took B16F10 cells. These are mouse melanoma cells, which are pigmented. And we treated with this small molecule UK5099, which inhibits the entry of pyruvate into the mitochondria. So we can decouple glycolytic and cytosolic metabolism from mitochondrial metabolism. And we can see that melanogenic genes like PMEL, M-LANA, and TYRP go down. we can see a decrease in the amount of melanin content. So essentially, these cells are losing melanin with no effect on growth. But we wanted to ask this in a more kind of comprehensive way. And so we teamed up with Steve Gigi at HMS to do quantitative proteomics. And here I'm just showing you that same idea. So we treated with either vehicle or this inhibitor of the mitochondrial pyruvate carrier and measured the abundance of all melanogenic genes. So we could see that the abundance of all of those proteins go down. And we can also do transmission electron microscopy under these conditions and basically see that the melanosomes themselves are perturbed. So we have less total melanosomes on the right and less in this mature state. And so what we want to suggest is that there's this generalized model that we kind of lucked into by trying to answer the question in a kind of fortuitous way. And so there are cells, progenitor cells, on the left in this kind of tan, I guess, that will eventually differentiate into this more mature cell. And along each step, there are lineage-defining transcription factors, some people call them master regulators, that set the tone, that tell that cell who they are, what to do, and who to be. And at the end of the day, you will get differentiated cells with specialized functions, right? So in skin, you have melanocytes. You have a specialized function to produce melanosomes, which make melanin, which gives skin pigment and protect you from UV damage. In erythrocytes and red blood cells, you will eventually eject the nucleus. You will lose the nucleus, you'll lose the mitochondria, and you're just a bag of floating heme that has to survive for 100 days in the human body, right? So you get very specialized functions. And it's known, well established, that these master transcription factors control that function. And what we want to add is that these master regulators also seem to set the metabolic state so that you can control metabolism to support that function, right? That specialization is encoded not just in who you are, but also the metabolism to make sure that you can do what you need to do. Okay, and then the last 10 seconds, I just want to say, I think there's a role for this kind of intermediate scale perturbation assay. It was a little bit nerve-wracking presenting this morning after Aviv and after Marion, because Perturb-seq is awesome. It's probably one of the coolest technologies that I can think of, but it's a little bit daunting, I think, as a cell biologist. And as a cell biologist, this is a common slide that people will see, right? You have cells, you grow them in a dish, you treat with a molecule, you treat with a compound, a ligand, whatever, and the first tool in your toolkit is often to do RNA-seq, right? See how the cells respond. And what we think is that by using troutfish to kind of understand the connection between your molecule and your output, you can really help understand mechanistic biology. And it's very cost effective, it's very easy. I think there's a mix of computational biologists and wet lab biologists in this room. I think all the wet lab biologists would laugh at how easy the protocol is, and the computational biologists would laugh at how easy the analysis is. But I think the most exciting part is I could have you swap roles, and you would each think it's still just as easy, right? There's nothing complicated about this assay. And for that reason and the low cost, we think it's a way to perhaps democratize and understand better for a general lab. Okay, with that, I'll say thanks, Jared, as my mentor, those that helped with the project, the Gigi lab, and then I didn't have time to show any of the data, but we've been working with Rich White to ask some of these questions in zebrafish as well. Thanks.
Questions?
That was very interesting. So do you think it's mostly a boosting? You know, what kind of levels are we talking about? Because you probably need, let's say, these glycolytic enzymes in every cell, so it's like a 2x improvement or 3x improvement. And sometimes you probably should see the opposite, not that you don't need this pathway anymore, and then your lineage transcription factor should repress it. Do you also see that?
Yeah, that's a great question. So I think specifically for glycolysis, as you said, almost all cells, right, in the human body will want to do glycolysis. What we found is that they kind of allow you to titrate, right? There's a basal amount of glycolysis that exists in every cell. That's what is required. But these lineage-defining factors kind of allow you to buffer, right? In those cells that you need, 2, 3x more in order to support certain needs like melanosome biogenesis and function, you're going to see that ability to buffer in these lineage-specifying transcription factors. And then we know that there's some cells that during the differentiation process may want to suppress glycolysis in order to engage in fatty acid oxidation, right? And so we can see that too, that there's kind of this suppression. But great question.
Great question from Zoom. To what extent can your finding be recapitulated or validated by using Weismann's PETA-seq data?
Yeah, that's a great question too. So we've looked a little bit for K562, and we can see, I don't know the numbers off the top of my head, but we can definitely see recapitulation in the K562 genome data set. Obviously, for the skin-dependent stuff, we can't see that, right? But that's a great question. We do think that there's pretty good correlation. When you can achieve genome scale per TRPseq, you can catch kind of this regulatory that we can catch with these assays as well.
Sorry, we have to stop here. Yeah. Thank you, Alex, for this wonderful talk.
Yeah.